Year one. Okay, hello everybody. Welcome to week 10 review. I'm Rosella.
John.
Okay. Okay. All right, I can just go like this. Oh yeah.
Okay. Polymerase proofreading, guys. As Laura was going over in class, it doesn't really make a lot of mistakes. Well, it can make mistakes, but it's really good at catching them and just fixing it right away because it has exonuclease function as well as polymerase function. And so there is a very, very tiny chance of it making an error and not catching it. So we're shooting with around 1 error per 10 million nucleobases. And so this happens because the polymerase has two different domains, and the exonuclease domain has 3 to 5 exonuclease activity. So if you can switch slides. I don't know if it's capturing my voice. I really hope it is.
I'm sure it is.
Yeah. So the polymerase theoretically is supposed to look like a hand, like this, with the fingers and the thumb. And while it's polymerizing / adding the nucleotides, the proofreading domain, which is the exonuclease domain, or which contains an exonuclease domain, is constantly checking for the newly added pairs, just to make sure that all the mistakes are being caught. So they're different domains, and that's the important part that you should take away from all of this.
Any questions? Okay, messenger repair. Okay, so DNA polymerase can proofread with its exonuclease activity, like John was talking about, but sometimes the proofreading does not catch all of the errors that the DNA polymerase makes, and that's when the mismatch repair system comes into play. And so basically, this mismatch repair is a post-DNA replication repair pathway that searches for and fixes bases that are mismatched, which means just like the incorrect complementary pair. And so an important thing in this MMR pathway is to be able to differentiate which is the template strand and which is the newly synthesized strand. And now we're going to go over why we need to correctly identify which is the template and newly synthesized strand. So over here, we have like 3 different scenarios. The first one is if there was no mismatch repair pathway. So if there was no mismatch repair pathway, and for example, an A got incorrectly paired with a G, this is the bottom strand is the template strand. So the original one with the correct base in it, and then the newly synthesized strand has the incorrect nucleotide. So this is the one that should get fixed, but there is no MMR pathway here. So what happens is that when this DNA then gets replicated again in another round of DNA replication, these 2 strands, the template strand and then the newly synthesized strand with the incorrect base, they're going to separate, and then DNA replication will happen. And what you're going to have is one set of DNA with the correct base pair, right? Because it still kept the G in the template strand, and then another one with a mutated DNA base pairing, because this A was not fixed in the MMR. And then this other scenario is if the MMR pathway would not be able to identify which strand it needed to fix. So for example, if it fixed the G instead of the A, what would happen is this G would then get turned into a T, so it would correctly base pair with A. And then when this DNA went through another round of replication and the strands separated, you would wind up with 2 mutated DNAs with incorrect base pairing. And so then this third scenario is what you would get with the MMR pathway, which is it sees that the A is the incorrect base pair because it knows that that is the newly synthesized strand. So it changes it to a C. And then once this DNA goes through another round of replication, you're going to get two unmutated normal DNA things. Any question about this? So therefore, this just shows how it's important to know which one is the newly replicated and which one is the template strand when doing MMR. Yeah, sure.
So can you go back to the slide?
Yeah.
So if you look at the DNA where the mutated region is on the original strand, or I guess on the newly synthesized strand on the first example, there is a circle around the A, and that's because when there is a mismatched pairing, the DNA has a lesion or just a distortion because the bases aren't correctly aligned. And so if you can look at also the second case, no, it's fine. If you look at the second case on the mutated strands post replication, there's no more distortion / lesion because it was assumed that the correct base was there. And the correct base for that pairing is also added. That doesn't necessarily mean that the process occurred correctly because you are fixing the mutated DNA in the wrong way. But it just goes to show that you only see these lesions if there's a mistake and it wasn't caught.
Okay, and then so different prokaryotes and eukaryotes have different ways of identifying which is the template strand and which is the parental strand. In prokaryotes like E. coli, during DNA replication, the DNA is hemimethylated, so the template strand has methyl groups at certain sequences. And then once the newly synthesized strand is made, there is like a certain amount of time where that newly synthesized strand isn't methylated, which is what basically gives proteins the ability to like sense which one is the newly synthesized strand. But then that newly synthesized strand, once given like a right amount of time, will also be methylated later on. So it's important to like catch the mistakes right after DNA replication. And then in eukaryotes, it's hypothesized that the way that the newly synthesized strand is identified is through the nicks in the DNA, which is the nicks that the DNA ligase needs to seal after replicate after the DNA polymerase polymerizes the strand. Okay, so this is just going over the mechanism of mismatch repair. So you can see that this bubble over here is where the mismatch happens. And then this is a nick in the DNA that the DNA ligase hasn't sealed after DNA replication. So what happens is that a complex of proteins basically takes the mismatch area and the nick, and then basically an exonuclease can come in and chew the newly synthesized DNA from the nick to the mismatch repair to the mismatched nucleotides. And so then, of course, since there is like a single-stranded DNA that's exposed, you need the single-stranded DNA proteins to help keep that template strand stable. And then because you have this, because you now remove the mismatch and then removed also a lot of the nucleotides from the nick to the mismatch, you have to resynthesize the strand, and DNA polymerase is going to come in and add the correct base pairing, and then ligase is going to come in and then seal that last nick. Oh, something I didn't mention is that helicase is needed to basically separate the nick, the template strand and the newly synthesized strand before the exonuclease.
And also with the visual, you're able to see that you need a lot of space in the genome to do this. And for a single nucleotide, you're wasting all the space, all these different resources. But it just goes to show how important making sure that the correct base pair is being added is.
Okay, and then this is just basically going over what I said about like how mismatch repair happens in eukaryotes. The eukaryotic one is on this side, and then the prokaryotic one is on the left side. Over here you can see the nick, and then that from the nick to the mismatch repair, get an exonuclease, choose back the DNA, and then over here is the sliding clamp so that the DNA polymerase can come in and replicate the DNA again. Over here, you have the single-stranded binding proteins to stabilize the template DNA while the DNA polymerase is replicating the newly synthesized strand again. And then you have ligase here that seals the last nick. Oh, oops. And then over here, you have the mismatch repair for prokaryotes, and then you can see this it can identify which one's the template strand and which one's the newly synthesized strand by seeing which one has the methyl groups. The strand with the methyl groups is the one that is the template strand. So therefore, the MMR machinery knows that it needs to remove a segment of DNA from the bottom strand because it's not methylated. So again, an endonuclease, which is different from here because it already has a nick. So an endonuclease needs to come in and make a cut in this newly synthesized strand. And then an exonuclease needs to go and chew back the mismatch DNA. And then DNA polymerase can come in and do DNA replication and then ligase. So an important distinction between like the prokaryotes and eukaryotes is that in the prokaryotes, you need that endonuclease to make a cut in the DNA before the exonuclease can come in and remove the DNA nucleotides, because in the eukaryotes, it already has that nick cut in the DNA. So that like it already like an opening for the exonuclease to come in. What's up? Yeah, that's that. Do you guys have any questions about the MMR stuff? Yeah.
One with it was like the slide above. So with eukaryotes, why
This one?
one more up. Like the neck, like I understand like the neck
Mm-hmm.
is needed. But like why is it so far away from like the DNA mismatch? Or is like
Oh, it's
kinda
random.
independent?
It's
Like
random.
the necks are just so they have to wait for like this random neck to happen before they're coming.
Well, remember when you're doing like DNA replication, the nick is wherever like
Wants to
yeah,
They
like
come-
between the DNA segments.
oh,
So do you remember like the Okazaki fragments? Yeah, so
Just
like
like-
between that, so it's sort of random because like
oh,
if
okay.
this is like if these are Okazaki fragments, this one and this is the other, like you could have a mismatch like right over here that would then need to use like a nick like
Oh,
here.
so, like wherever it
or-
ends,
Yeah,
okay.
exactly. So it'll just use, I guess, like closest one or whatever.
Okay. And it has
Well, also
to go
because
through the food.
It has no, so you're not expecting to have any of the mismatches,
Yeah.
right? So they're like,
They're just kind of going.
they're theoretically not supposed to be there. And because the Okazaki fragment is only so big, you don't really have like a way to determine, oh, I need a nick here. It's
Mhm.
not just happening.
And it needs to go from the, is it like the 3'? That's the start on the 3' side? For
The exonuclease activity, yeah.
Okay.
So choose that.
So it needs to go from like the 3 to the 5, so it's 2 towards the 5. Okay,
Yes.
thank you.
Good, good. Alrighty, I
And
think I'm
that's
good.
creating a new 5' to that you
Should I
So
stop?
like the new, we say it's like creating a 5', so it's like chewing this way.
and then there's a space available here, and then it's going that way again. And then it's going the opposite way.
So then it chews this,
You choose,
and it goes
and then
to a
you need to
certain point.
fix it back the opposite way.
So it's exonuclease, but then it doesn't fix it, does it? Or is that
The polymerase?
the
Should I stop
polymerase, not
recording?
the-
Yeah.
Alright, bye guys, thank
I
you.
know the polymerase like fixes it, but once the